Hello, my name is Alan Brown, and today I'll be discussing my integrative analysis of histone modifications, transcription factor binding, and gene expression in Plasmodium falciparum. So I'm going to jump to this slide. Plasmodium is a single-celled eukaryotic parasite with a very complex life cycle. It starts when an infected mosquito bites a human. The sporozoites invade the liver. After a period of development, they burst out into the bloodstream and start the asexual blood cycle. Some small percentage of these commit to the sexual phase of development, develop in the bone marrow, and then can be picked up by a new mosquito and spread to other humans. So this is a very complex life cycle with many changes and many shifts in gene expression, but very few transcription factors. So since something is regulating their genes and moving things around, this study is looking at transcription factors and their association with chromatin, like via chromatin states. So we have data mainly from the asexual blood stage. This is broken up into three main parts: the ring stage, trophozoite stage, and schizont. This is important because I have data from all three of these stages. This is a 48-hour cycle. So my data for the ring stage is at like 10 hours post-invasion. I have data from 30 hours and 40 hours. And I'm making chromatin states from these data sets. This is an example, and then this panel 3, this is an example of some of the marks and ataxic and histone variants that went into calling the chromatin states. So this is just showing the tracks for the 40-hour post-invasion, but I did use 10 hours, 30 hours, and 40 hours together to create these state definitions. And it gives us a useful categorical variable for thinking about this. So this is an important figure for interpreting anything else with the chromatin states. I have my 11 states sorted by the emission probability for ataxic, and that this is open chromatin is just the name of this. These emission probabilities come from Chrome HMM, which created the model for this, and this panel next to it shows you the percentage of the genome covered by each state at each time point. Because remember, I have a ring, troph, and schizont, 10 hours, 30 hours, and 40 hours post-invasion data sets. This is also the association of genes to each state at each time point, like a count for the genes that have this state primarily in like the early beginning of the coding sequence, some part of the UTR, the 5' UTR. I can talk with you about all of this, but 5 minutes goes by fast. So this is gene expression data, the average gene expression for genes where this state is in their transcription start site, and another like feature enrichment matrix here showing how these states overlap different parts of the genome. Like a combination of all of this information is how I named some of these states. Okay, moving back up to panel 4. This is some of the focus is looking at chromatin states at transcription factor binding sites. So for this, I picked 3 examples: AP2LT, which is correlated with active gene expression, so an increase in gene expression at downstream genes. It's expressed around the 30-hour post-invasion mark, which is why we see the number of peaks going up. That's related to seeing the number of peaks increase. And you can see this is chromatin at the binding sites for any of the peaks at any of the time points. So it's like the superset. So you can see that the chromatin under binding sites is shifting towards more active and less H2AZ. Next, we have AP2G3, which is an important regulator for the sexual phase of development. Most of these genes are turned off during the data, like the life cycle stage that I have. And the chromatin is more stable for this set, but this is a similar pattern for several of my, they don't mind, but several of the sexual phase transcription factors. CYP2 is an important regulator, and it's often associated with telomeric regions and sort of some chromatin, chromosome folding and creating different boundaries. So this HP1 state is very common at the ends of chromosomes and in centromeric regions. Last, I would love to talk about this more in person. This is an enrichment plot showing for each state their enrichment compared to background of a particular transcription factor that was chipped at a particular time point. So I have factors chipped at many different time points throughout the life cycle. A couple of quick things to note as I'm running out of time. The quiescent stage covers like 30% of the genome, but it's much less likely to occur at these states. You can see some patterns like AP2LT in the 10 hours, and then the 30 and 40, there's a low level of ATAC-seq or chromatin accessibility, and that increases over time. There's a block of states that have this like G mark related to gametocytogenesis, the sexual stage, where they're associated with some level of AKT9-trimethyl, HP1, which are repressive marks, but present in gene bodies. So maybe these are having a repressive role, holding down, knocking down sections of the chromosome. Please come talk to me and have a good conference.